Hi everyone, welcome to my channel Pure Biology, and today I come with another video, that is the WOBBLE hypothesis. And WOBBLE hypothesis is related to the translation process, and this translation process and genetic code I have already explained in my previous videos, so you just check out that. And here I try to explain about the WOBBLE hypothesis. What is that? It was first proposed that a specific tRNA anticodon would exist for every codon. So one anticodon that exists for one codon. Okay, that means these are the complementary. One anticodon of the tRNA exists for a codon. Okay, and codon obviously triplet. And if that were the case, at least 61 different tRNAs, possibly with an additional 3 for the chain terminating codons would be present. So these are already explained that how much number of terminating codon of the stop codons present, and also the initiation codon is how much number present. So here it is saying that at least 61 different tRNAs or anticodon that present, and which has a complementary codon. Okay, so WOBBLE concept, this is one opinion. This is the opinion of Francis Crick. Okay, Francis Crick and that is the WOBBLE concept according to Francis, I think Frick or Crick. One second. Francis Crick or Frick, I have to say first. Okay, so the scientist name is Francis Crick. According to Francis Crick's hypothesis, that is the WOBBLE concept, the base at the 5' end of anticodon also shows non-standard base pairing with any of several bases located at 3'. What is saying that 5' simple way, this is the 5' end is attached with the 3' end. See here, this is the rough diagram. You can see this is 5' to 3', and the 3' that is coded with the 5' codon. This is the anticodon that present with the tRNA, and this is the codon that present in the mRNA. So 5' and 3', these are the related. Again, this is the 5' and 3' here. Okay, so this is the thing, and the first base of anticodon and the third base of codon. Okay, I just was checking this one is not seen from the video or from the camera, not video, from the camera, so that's why I first have to check that it is seen or not. Okay, so this is the these are the rough diagram where you can see these the tRNA anticodon is that one, and the mRNA codon is this. So what is saying that first base of anticodon and the third base of codon is the WOBBLE position. What is saying that first base of anticodon, that means this one, and the third base of the codon, and the number starting in case of anticodon from here to this, and in case of codon starting from here to this. So this is the third place of the codon, and this is the first place of the anticodon. Clear? These are the WOBBLE. Okay, so here if G is present, that means there is a chance of binding C or U. And in second case, if there is A, so there is if there is U, so there is a chance of binding A and G. If there is I, so there is a chance of binding A, C or U. Okay, so another two the bases are obviously there, must be there, but this place is the third place, and this place is the first place. So if the first place of the anticodon is I, that means a chance of third place of binding as codon if as A, C or U, and like that this type of U, C like that, or U, A, U like that. Okay, so but the position is the third position, that may be U or C or A. Clear? In case of if in that position if the G is present, so there is a chance of binding C or U. Okay, another nucleotide bases are obviously there. So in this is also another same thing happening. Okay, so this is known as the hypothesis of WOBBLE hypothesis. Okay, and there are 20 amino acids present, and the name of these amino acids obviously histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, valine, alanine, and asparagine and arginine, aspartic acid, cysteine, glutamic acid, glutamine, glycine, proline, serine, and tyrosine. Okay, so these are the 20 amino acids. So here this is very simple topic, that is the WOBBLE hypothesis. So if there is any problem to understand how this binding of codon or anticodon is occurring, so you have to understand this WOBBLE hypothesis, because without understanding of WOBBLE hypothesis you cannot understand that how that more than codons that can bind the same amino acids. Okay, so this is possible due to the degeneracy of codon, and another thing is the WOBBLE hypothesis. Clear? So I will try to another topic. Okay, another thing I have to explain here. First I have to wrap this and then I try to explain with you. Okay. Okay, here I will try to explain the code dictionary, how the code dictionary is occurring. Okay, here I have to write that UCAG, and here is a UCAG also. Now just multiply them. How is that possible? You can see U and U present in this area. So first codon, that must be U, U and E. Second one is what? Second one is U, U and C. So U, U and C. Okay, sorry. Okay, so second is that UUC. Third one is that UUA. UUA. And the fourth one is that UUG. UUG. In case of C, what is happening? U, C, U. U, C, U. Second one is that U, C and C. U, C, C. Third one is that U, C, A. U, C, A. Fourth one is that U, C and G. Clear? Next column. Next column is that what? U, A, U. Then U, A, C. Then U, A. Next U, A, G. Clear? In the next column, U, G, U. Next U, G, C. U, G, A. U, G, G. Why U is always in the first tab? Because this is the horizontal rows of U and column U, C, A, G. Clear? In the second, that horizontal column, horizontal row is C and the column is U. So how do you write? C, U, U. Next C U C. Next C U A. As this is the column of U, that's why we have to write middle, in the middle U. You can see. Next C U G. Okay, this is C U, then C and C. Next C A, and here C G. So how we write? One U from here, one U from here, then this is present in U, that's why U. And if you see this one, then one U from here, one U from here, and this is the column of the C, that's why C is written in the middle. Okay? And if you can see here, this is a row of C, so all the first nucleotide base, that is the C. So C is there and column is U, so that's why written with U. CU for that, one for horizontal row, another for column, and then this is starting from U, then starting from C, and then A, then G. Clear? Now here this A, what will happen here? C A U, C A C, C A and C A G. Okay, this is for, what is this? This is for this horizontal C, this is for this column C, and then U C A G for U, for C, for A, for G. This is the U C A G. Clear? Next C G U, C G C, C G A, C G G. Okay, C for this and G for this, and then U C A G. Okay, sorry. Next A U, then A U C, then A U A, then A U G. Okay, next A C U, A C C, then A C A and A C G. Next A U, A C A and then A G. Next A G U, A G C, A G A G G. Last one is that G. Okay. G U G U C, G U A, G U G, G C U, G C C, G C A and G C G. Next G A U, G A C, G A and G A G. Last one G U, G C, G A and G G. Okay, this is for phenylalanine, this is for leucine, this is for serine, and here this is for tyrosine, and this is for stop, and this is cysteine, and this is stop, and this is TRP, tryptophan. Next, this is leu, this is pro, this is HIS, this is GIR, this is ARG, this is isoleucine, ILU, this is the methionine, and then threonine is that THR, this is the ASR, this is the LYS, this is serine, and this is arginine. Next, valine. Okay, this is alanine. This is aspirin, GLU. This is glycine. Okay, UCAG, 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 and UCAG. So this is the code dictionary. Okay, so I think you can understand how the code is formed. So here these are the horizontal row, and these are the column row. These are the column. And then how do we count? You just write down one horizontal and one column, and then in the third position, or you can say the first position, this always remain UCAG. Simple way. Another example I have taken. This is the C. So this is the column of C. So and row is U. So U is written in the first, and column C is written in the middle, and last one is always UCAG. So in that way, if you can take any example from here, if we take this one, so here you can see one is the horizontal A, and one is the column C, and last one is the UCAG. So in that way, all the codons generally form. Okay, this is a code dictionary. So I think you can understand it properly, how the codon is formed and how the WOBBLE hypothesis is important for the transcription process or the binding of anticodon and codon. I have already explained here. Okay, if this is helpful to you and you are the newcomer in my channel, obviously share and subscribe this channel, and this